Hello everyone, welcome to the new session of Genetics and Plant Breeding, Cell and its Structure. So this is the fourth part of this chapter. Okay, so we are going to study about three different organelles in this session, that is mitochondria, chloroplast and nucleus. So let's begin with the mitochondria. So mitochondria is a very important organelle, okay, but they cannot be seen. They cannot be seen under the microscope unless they are stained, unless they are specifically stained. So if we want to see the mitochondria under the microscope, we have to stain the cell. Okay, the number of mitochondria per cell is variable depending on the physiological activities of the cell. So the number of mitochondria varies from cell to cell. Some of the cells are physiologically very active, okay, physiologically very active, like our heart muscle cell, that is, they are myogenic. Okay, so they are continuously working, they are continuously pumping the blood, so they require lots and lots of energy. So their muscle cells are having large number of mitochondria. Okay, so depending upon the requirement or the physiological activities of the cell, the number of mitochondria varies. In terms of shape and size also, there is a considerable degree of variability is observed in mitochondria. Some mitochondria are in this shape, some are a round shape, some are in this discoidal shape. So there are different shapes of mitochondria and also of different sizes. So size and shape varies in the mitochondria. Okay, typically a sausage shaped or cylindrical, having a diameter of 0.2 to 1 micrometer average, that is 0.5 micrometer, and length varies from 1 to 4.1 micrometer. So this is the particular size, okay, size of the mitochondria. Mitochondria is a double membrane bound structure, very important. So if you see, there is an outer membrane and there is an inner membrane. So in between these two membranes, first and second, there is a compartment. So if I draw a structure of a mitochondria, cross section of a mitochondria, then we are having structure like this. So these are the invagination of the inner membrane of mitochondria, like this. These are forming invagination, and that invagination is called as cristae, right? So this is the outer membrane, and this is the inner membrane. Now, in between this outer inner membrane, there is a compartment. Right? Compartment. Outer compartment, and this is also one compartment, that is called as inner compartment. Right? So this is very important. So mitochondrion is a double membrane bound structure with outer membrane and the inner membrane, dividing its lumen distinctively into two aqueous compartment, that is the outer and inner compartment. So here we can see we are having two compartment. One compartment is between the outer and inner membrane, and the another one that is very important, that is the inner compartment, right? The inner compartment is filled The inner compartment is filled with the dense homogeneous substance called as matrix. So here, this green color which is shown here is the matrix. Okay, that is the fluid. It is a dense fluid. Okay, and that is called as matrix. The outer membrane forms the continuous limiting boundary of the organelle. So it is quite smooth. Outer membrane is quite smooth, and that is the limiting boundary of the mitochondria. Whereas the inner membrane invaginate, it is not smooth, okay, and form a crystal-like structure. So here comes the inner membrane forms a number of infolding or invagination called as cristae, right? Towards the matrix side, The cristae increase the surface area. Yes, if there is a folding of membrane like this or invagination of the membrane like this, then this increases the surface area. Right? So cristae increases the surface area. The two membranes have their own specific enzyme associated with the mitochondrial function. So their membrane, this membrane, that is the outer membrane, right? Outer membrane, they are having some specific proteins, right? Specific proteins that are present in the form of enzymes, right? So outer membrane is having some specific enzyme. In the same way, inner membrane are also having some specific enzymes attached to it. So enzymes are proteinaceous in nature. So these two membranes are having specific enzyme attached to their corresponding functions. Mitochondria are the site of aerobic respiration. Yes, very, very important. Aerobic respiration takes place in mitochondria, whereas anaerobic respiration takes place where? It takes place in cytoplasm itself. Okay? Mitochondria, they produce cellular energy in the form of ATP. So with the help of anaerobic respiration, lots and lots of ATP is produced inside the cell, right? So that is the source of energy in living body. Hence, they are called as powerhouses of the cell. So this particular organelle is also called as powerhouses of the cell. The matrix also possesses single circular DNA molecule. Very, very important. So if we again see the structure of a DNA, okay, so this is the outer membrane. Now I am drawing the inner membrane. Okay, inner membrane are folded like this. Okay, so this is the inner membrane. Yeah, and this is filled with the matrix. Like this. So this is the matrix, and this matrix is having DNA molecule. This is having DNA molecule and few ribosomes also, Now these ribosomes are of 70S type. So we have earlier seen that 70S type of ribosome is a peculiarity of prokaryotes, right? So this prokaryotes is having ribosomes of 70S type. That 70S type ribosomes are present inside the mitochondria, whereas the eukaryotic cytoplasm is having 80S type of ribosome. Very, very important, right? So matrix also possesses single circular DNA molecule. A few RNA molecule ribosomes are present, that is of 70S type, and the components required for the synthesis of protein. So some proteins are synthesized inside the mitochondria with the help of 70S type of ribosomes. Okay, so our next organelle is the plastid. So plastids are found in all plant cells and in euglenoids. So euglenoids comes under the protista. We have already seen that it is present in the protista. So they are easily observed under the microscope as they are large. Okay, so they are quite large in size, so they can be easily observed under the microscope without any staining. They bear some specific pigments and thus imparting specific color to the plants, right? So they can have chloroplast, chromoplast, and leucoplast, right? So based on the type of pigments, the plastid can be classified as the chromoplast, chloroplast, or the leucoplast. Okay. The chloroplast contain the chlorophyll pigments and carotenoids pigments, so which are responsible for the trapping light energy essential for the process of photosynthesis. So these two pigments are present in the chloroplast. These two pigments are present in the chloroplast. Chromoplast. Now, they are fat soluble carotenoids pigment. They are fat soluble carotenoid pigments like carotene, xanthophyll, and some other pigments are also present. Okay, but basically these two are the important one in the case of chromoplast, that is the carotene and xanthophyll. So these pigment provides color to the flower, like they can be red, they can be yellow, orange, white, right? So these type of flowers of different than green color, they are because of the chromoplast. They give parts of the plant a yellow, orange, and red color, right? Leucoplast, the third one is the leucoplast, are the colorless plastid. They do not have any pigment of varied shape and sizes, and they store nutrients. Instead of presence of pigments, they are having stored nutrients in it. They can be amyloplasts that store carbohydrates in it, for example starch, right? Example in the case of potato. Elioplast that stores oil, that store oils in it, and fats, whereas the aleuroplast that stores proteins in it. So there are three different types of leucoplast: amyloplast, elioplast, and the aleuroplast. So amyloplast stores carbohydrates in it. Okay, elioplast stores oils in it, and aleuroplast stores proteins in it. Okay, so this is about the leucoplast. Majority of the chloroplast are green plants and are found in the mesophyll cells of the leaf. So we are talking about the chloroplast. Okay, so majority of the chloroplast are present in the green parts of the plants, and if we see the mesophyll cells of the leaves, right, they are present in abundant. Okay, these are lens-shaped, oval, spherical, discoid, or even sometimes ribbon-shaped. Sometimes in the form of ribbon-shaped. Okay. And they are of variable length, like their length varies from 5 to 10 micrometer and width is around 2 to 4 micrometer. The number varies from 1 per cell to the a green alga, that is chlamydomonas. They are having just 1 per cell. The chloroplast is 1 per cell. Okay, and 20 to 40 per cell in the case of mesophyll cells of the leaves. Okay, so like mitochondria, the chloroplast are also the double membrane bound. So if we see the inner structure of plastids, then we see that like mitochondria, they are also the double membrane organelle. So here you can see outer membrane and there is an inner membrane, outer membrane and inner membrane. So of the two, the inner chloroplast membrane is relatively less permeable. It is relatively less permeable, but the outer membrane is highly permeable. Okay. The space limited by the inner membrane of the chloroplast is called as the stroma. So if we see the inner structure of the chloroplast, then we are having outer membrane and inner membrane. This is the outer membrane and inner membrane. Inner membrane. And this compartment, the inner compartment, is filled with the liquid substance that is called as stroma. That is called as stroma. Okay. Okay. A number of organized flattened membranous sac called thylakoids are present in the stroma. So here you can see a thylakoid-like structure stacked over one above the other. It is placed like this, one above the other. Okay, so these stack like structure, like if you put a coins, number of coins one above the other, then it form a pile of coin like this, right? So there are number of these thylakoids pile structures are present, and sometimes they are also attached with like this. So this you can say a sac a stack of thylakoid is called as grana. It is called as grana. Okay, and this two grana are sometimes attached with the lamellae structure that is called as stroma lamellae. Stroma lamellae, right? Okay, so thylakoids are arranged in a stack like the pile of coins and they are called as grana. Okay, or the intergranal thylakoids. Okay, so these are the grana structure. They are very, very important for the process of photosynthesis. Okay. In addition to their flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. So we have discussed that different grana are attached with the stroma lamellae. The membrane of the thylakoids enclosed space called the lumen. So there is, if we draw a one thylakoid, then there is the lumen. There is the lumen inside this thylakoid structure. The stroma of the chloroplast contain enzyme required for the synthesis of carbohydrates and proteins. So we are having stroma, the liquid parts, the matrix parts of the chloroplast. Okay, so they contain lots and lots of enzyme concerned for the synthesis of carbohydrates and protein. It also contained the double-stranded circular DNA, like the DNA is present in the case of mitochondria. So chloroplast also contain double-stranded circular DNA and ribosomes. Here also the ribosomes are 70S type as it is present in the case of prokaryotes. So chlorophyll pigments are present in the thylakoids. Okay, these pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller, right? 70S than the cytoplasmic ribosome, that is 80S in the case of eukaryotes. Okay, so third organelle is the nucleus. So nucleus is, as a cell organelle, was first described by the Robert Brown. Okay, the first time it was described by the Robert Brown in early 1831. Later, the material of the nucleus stained by the basic dyes that gives the name chromatin by the flaming. So inside the nucleus, there is a material that came to know after staining with the basic dyes. Okay, so after the staining with the basic dyes, we came to know that there is some material present inside the nucleus. What is that material called? That material is called as chromatin, and it was the name given by Fleming. The interphase nucleus. So we are having a cell cycle. Inside the cell cycle, we are having some phases like interphase, the nucleus of a cell when it is not dividing. So when the nucleus is not dividing, then we can say that the nucleus or the cell is in the interphase stage. Okay, so when the cell enters to the division phase, then it comes the S phase. Okay, G1 phase, S, G2 and M phase. Okay, but when the cell is not in a dividing phase, then it is present in the interphase. Okay, so as highly extended and elaborate nucleoprotein fibers called chromatin. Okay, so in the interphase, the chromatin material is present. So when the cell is in interphase, we can see one thread is present and it is very long, and that is called as chromatin material. Chromatin material. Nuclear matrix and one or more spherical bodies are called nuclei. So the liquid substance present inside the nucleus, that is called as nucleoplasm. Nucleoplasm or nuclear matrix. Nuclear matrix, right? And some spherical bodies are also present. So that is called as nucleolus. That is called as nucleolus. The outer membrane usually remains continuous with the endoplasmic reticulum and also bear the ribosomes on it, right? So outer surface of the nucleus, they are continuous with the endoplasmic reticulum, and some ribosomes are attached to the outer surface. Okay, so this is the nucleoplasm, that is the liquid substance present in the nucleus. Nucleolus, the round structures. Okay, here is the round structure which is present inside the nucleus, that is called as nucleolus. Nuclear pore. Now there is a small gap between the membrane of the nucleus, that is called as nuclear pore, and there is a nuclear membrane, that is a delimiting membrane of the nucleus. Electron microscopy has revealed that the nuclear envelope, which consists of the two parallel membranes with a space between 10 to 50 nm, called the perinuclear space. So again, the nucleus is a double membrane. Okay, and there is a space in between the outer and the inner membrane, that is called as a perinuclear space, and forms a barrier between the material present inside the nucleus and to the cytoplasm. Okay, at a number of places, the nuclear envelopes is interrupted by the minute pores. Okay, so here comes the minute pore. So if this is a nuclear membrane, then there is a minute pore present like this. So this is called as nuclear pore. Okay, so which is formed by the fusion of its two membranes. These nuclear pores are the passage through which the movement of RNA and protein molecules take place in both the direction between the nucleus and the cytoplasm. So we all know that after the division of the DNA, okay, the DNA divides equally, and that is the process of replication. Now on the DNA, RNA gets synthesized, that is the process of transcription. Okay, but the protein gets synthesized where? In the cytoplasm. So this RNA is a site of protein synthesis. So RNA comes out of this nuclear pore to the cytoplasm, and in outside the cytoplasm, so their ribosomes are present, so synthesis of protein takes place in the cytoplasm, but the synthesis of RNA takes place inside the nucleus. So RNA comes out of this nucleus through these nuclear pore. Okay, normally there are only one nucleus per cell. Variations in the number of nucleus are also frequently observed. So many cells where we can see that there are more than one nucleus. Some mature cells even lack nucleus, like RBC, erythrocytes of many mammals, and the sieve tube cells of the vascular plant. They do not contain any nucleus. The nuclear matrix or the nucleoplasm contain nucleolus and chromatin material. This we have already seen, that they contain the chromatin material and the nucleolus. Nucleoli are the spherical structure and present in the nucleoplasm. So these nucleoli are present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm, and it is not a membrane bound structure. So nucleolus is not a membrane bound structure, and it is a site of active ribosomal RNA synthesis. So RNA synthesis take place where these nucleolus structures are present, and they are non-membrane bound structure. Okay, so larger and more numerous nuclei are present in the cell, actively carrying out the protein synthesis, because for the protein synthesis we required lots and lots of ribosomes. Okay, so ribosome are present, synthesis take place where there is nucleolus. So more number of ribosomes are required. Okay, so more number of nucleolus are there. So larger and more numerous nuclei are present in the cells, actively participating in the protein synthesis. Chromatin contain DNA and some basic proteins that are called as histones, and some non-histone proteins and also called as the RNA. So these are the basic proteins that are called as histones, and they are very important for the synthesis of chromatin material. Okay, they are attached with the DNA, and also some non-histone proteins along with some RNA material, right? So chromatin material is made up of DNA, some basic proteins, some non-histone proteins, and also the RNA material. A single human cell has approximately 2-meter long threads of DNA distributed among its 23 pairs of chromosomes. So if we join all the chromosomes in the form of a chromatin material, then it is 2-meter long thread like structure. So every chromosome visible only in the dividing cell essentially has a primary constriction or the centromere. So here we can see this region is the primary constriction, showing the primary constriction. Okay, so on the sides of which disc-shaped structure called as kinetochores are present. So at this particular region, a disc-shaped structure is present which is called as kinetochore. Centromere hold 2 chromatids of a chromosome. So two chromatids are present in a one single chromosome. So they are held by the centromere structure. Based on the position of the centromere, the chromosomes can be classified into four different types. So where the centromeres are present, on the basis of that, the chromosomes are of four different types. Okay, so let us see what are that. Metacentric chromosomes. So here this is the metacentric one, in which the two equal arms of the chromosomes are there. So equally, this arm and this arm is equal in size. Okay, and the centromere is present in the middle. It is present in the middle. So that is the metacentric chromosome. So this is the metacentric chromosome. Okay. Submetacentric chromosome. So centromere is slightly away from the middle of the chromosome. So here we can see it is not present in the middle. One arm is shorter and the other arm is longer. This is the short arm and this is the long arm. So this is called as submetacentric. Right? This one, the first one was the metacentric, in which both the arms are equal in size, right? In the case of acrocentric chromosome, the centromere is situated close to the end, forming one extremely short and one very long arm. So here we can see very short arm. This one is very short, and then this one is comparatively quite longer one. So this is the acrocentric. This one is the acrocentric chromosome. Okay. Sometimes a few chromosomes have non-staining secondary constructions or at a constant location. Okay, so whereas in the case of telocentric chromosome has a terminal centromere. So in the case of this one, telomeric chromosome. Okay, so there is a terminal centromere. In this case, there is a terminal centromere. Sometimes in a few chromosomes have a non-staining secondary construction at a constant location. Sometimes instead of having one construction, they are having secondary construction also. So this gives the appearance of a small fragment called the satellite, right? Okay, so this is all about this session. So we have discussed in this session about the mitochondria, about the chloroplast and the nucleus. So we will have one more session to complete the cell and its structure part. Okay, till then, thank you very much.